So let's do the continuation of the formation of notochord. So here as you can see that this is the notochordal process. So here and this is the blastospore or the cavity of the primitive node. And here this is the hypoblast. Now here from this to this you can see here basically that in this primitive node proliferative cells are present. And also and this is the blastospore, and now here in this diagram you can see here basically that this cavity of blastospore so here extends into the notochordal process. So here this is the notochordal process and this cavity, so here this is the blastospore and this is the cavity, and this cavity of blastospore as you can see here extending into notochordal process, and this leads to formation of notochordal canal, and that portion I have written here. So simply as you can see that the cavity of blastospore extends into notochordal process to convert notochordal process into a tube, basically notochordal canal. You can simply see in this diagram. So here that this is a notochordal process and here and this is notochordal canal. So here basically difference is that the extending of the blastospore cavity into the notochordal process. Now here you can see here that this is a notochordal canal and this is the epiblast and this is the hypoblast. Now here you can see here basically that till now that these are the so here basically a notochordal canal cells, so here don't mix with the hypoblast. Basically these are the cells forming the floor of the notochordal canal. They are separated they are separated from the hypoblast cells, but here you can see here that these floor forming cells of the notochordal canal so here mixed with the so here hypoblast cells. So basically I have written mixing of cells forming floor with hypoblast. So therefore I have written that the cells forming floor of the notochordal canal are mixed with the hypoblast cells. But here you can see here that till now basically all the cells forming the floor of the notochordal canal, they are continue with one another. As a result, basically no cavity nor any space found between any two cells. So therefore I have written that, but the floor forming cells, they are joined together so that no cavities. And also you can see here that this so due to this no space no cavity, you can see here that this notochordal canal is not connected with this primary or the yolk sac. So therefore I've written basically the floor forming cells separating the notochordal canal from the yolk sac because there is no space. But here in this diagram you can see here that this is a notochordal canal. Now here also that is still now all the cells forming the floor, they are so here joined together, so therefore no cavity nor space found between the cells, and but they are joined or mixed with the hypoblast cells. But now you can see here that slowly that is cavities or spaces begin to appear among the so here cells forming the notochordal canal. So as a result basically that is a continuation so here occurs. So therefore the cells forming the floor of the notochordal canal so here basically break down so that small cavities appear among the cells. And here and later you will understand that, and this will in future lead to the formation of notochordal plate. I have written, I'm going to explain this also. So here also basically you can, this is the epiblast, hypoblast, and this is the yolk sac. So therefore cavities appear as a result. So therefore connection made. Now here you can see here that initially that in the notochordal canal basically it is a rounded shape, and walls they are not flattened, they are rounded. But here you can see here basically walls of the notochordal canal have become flattened, and also basically cavities are there to ensure connection of this notochordal canal with the yolk sac. Now notochordal canal, when the walls become flattened and also and it has connection with the yolk sac, now the notochordal canal leads to formation of notochordal plate. So therefore written that this leads towards the formation of notochordal plate. So therefore I written finally when the walls of the notochordal process become flat, so that the shape changes from a round shape to a flat shape. Now the notochordal process called the notochordal plate. Now here you can see here basically that this notochordal canal is connected to the yolk sac, and in the beginning also you can see here that is notochordal canal. So here that is notochordal canal also connected with the amniotic cavity through this blastospore. So through this blastospore, so here a notochordal canal connected with the amniotic cavity, and here basically through these cavities, so here notochordal canal, so here connected with the yolk sac. So therefore very important portion I have written there. So here now the notochordal canal communicates with the yolk sac and also amniotic cavity through blastospore, and this leads to, so here, diffusion of nutrition from yolk sac to epiblast. So friends, now here you can see here that this is the notochordal plate. Now here, yes, the flattening happens, flattening of the notochordal canal happens, but again it converts into a rounded solid tube, and basically, and this leads to formation of notochord. So here is written that the flattening of the notochordal canal is reversed, and again the notochordal canal assumes the shape of a tube, and also basically an inductive proliferation of the cells occurs, and as a result, so here notochord formation occurs. Due to proliferation of the cells of the notochordal process, the tube converted into a solid rod of cells, and this is called notochord. If you see in all the diagram that is notochordal now, so first that as you have understood that from this notochordal process, that this notochord form, now that this notochordal process always lies in the midline, so therefore that this notochord also, so here lies in the midline, and also that is in future that this notochord will be replaced by this vertebral column, and that is, and its remnant will be known as nucleus pulposus. That's all about notochord formation. So first of all, basically notochordal process form, then notochordal canal form, then notochordal plate form, and finally, so here notochord form. That's all about notochord formation and the series. Now I'm going to discuss here about the formation of germ layer. So first of all, that these are the epiblastic cells. This is primitive streak, and this is a space between epiblast and hypoblast, and this is the hypoblastic cells. And here you can see here basically that is yellow colored are the hypoblastic cells and pink color are the epiblastic cells. Now here in this diagram you can see here basically that epiblastic cells are undergoing, so here basically proliferation, and through the primitive streak they are moving downward towards the hypoblastic cell layer. Now here you can see that finally that this hypoblastic cell layers replaced by this epiblastic cells. Now here, and due to this replacement of hypoblast by the epiblastic cells, endoderm form. See, the epiblastic cells arising from the primitive streak displaced the hypoblast to form endoderm. Now here that on the same basis, due to proliferation, that is movement through the primitive streak, that is epiblastic cells move into the space between the epiblast and hypoblast. So here, and then also basically mesoderm formation occurs in this way. So here that this is the space between the epiblast and the hypoblast. Now here, and in this space also basically epiblastic cells move and they lead to the formation of mesoderm. So similarly the epiblastic cells from the primitive streak go in the space between the epiblast and endoderm to form an intraembryonic mesoderm. Now here you can see here that finally that these epiblastic cells also undergo modification to form a new cell layer, basically ectoderm. Now here basically three germ layers: ectoderm, mesoderm, endoderm, and also primitive streak form. So here basically I have written the remaining epiblastic cells modify themselves to form ectoderm, and also basically you can see here that these three germ layers formed and primitive streak form, and the formation of the a primitive streak and the three germ layers leads to a process, and that process is known as gastrulation, which is our main topic. So that's all about it. So here, so first of all, that for the formation of primitive streak, so first of all, basically procaudal plate is required, and procaudal plate helps in the formation of primitive streak, and also that for the formation of notochord. That's all. Thank you.